Good afternoon, everyone. Welcome to our 12th session of the Elevator Pitch series. Today, the session will be conducted in English as requested by the organizers. And we have a very interesting session today from Sara and Julia from the Center for Genomic Regulation in Barcelona. So, María, Mayán, when you want to present our speaker.
Okay, hello everyone, and hello Sara. I just would like to thanks to Sara and to Julia to organize this elevator pitch, and I'm just going to make a short presentation of the trajectory of Sara, which is an excellent trajectory. I just will make some remarks. Sara is leading her research group in the Center for Genomic Regulation in Barcelona. She obtained her degree in biotechnology in the University of Florence and the PhD in Barcelona in the Institute for Research in Biomedicine. And she moved to Vienna to do her postdoctoral stay since 2013 to 2018, and she joined to the CRG in Barcelona in 2019 as PI, and also she's leading a ERC starting grant. Among her work, it's important to remark that she described physical interactions between metabolic enzymes and chromatin, and she characterized the function of some of these enzymatic activities, which are necessary for the maintenance of transcriptional programs that favor tumor growth. She has published more than at least 15 publications in journals such as Nature Genetics and Nature Metabolism, and she also registered 2 patents. And among the top awards, we would like to remark that she is a ERC grantee. Thank you very much, Sara. And now you just can start your presentation with Julia. Thank you very much.
Thank you, Maria. Yes, today, along with Sara, we have our junior researcher. Today will be Julia. Julia is a predoctoral researcher at the Center for Genomic Regulation and the Pompeu Fabra University. In 2019, she finished her undergraduate studies in human biology, during which she made a stay at the University of Linköping in Sweden. In 2020, she graduated from the Master of Multidisciplinary Research, after which she obtained an F.I. Agaure scholarship to continue with her doctoral studies. Currently, her research focuses on understanding the metabolic essentialities in lung cancer and their differences in some hyperaggressive types. So, Julia, thank you so much for moderating this session. And whenever you like, the floor is yours.
Okay, so our first speaker today, it's going to be Sara Sdelci, that was very kindly introduced by Sara and Maria. So Sara, whenever you want, you can start your presentation.
Okay, so you can see the presentation and you can see the screen, right? You can see myself as well, right? Yeah, okay. So thank you very much, Julia, and Maria and Sara for the introduction. Yes, okay, so there is some connection problem maybe?
Yes.
Okay. No, everything's good. You can.
Is everything good? Ahead. Okay, because I don't see anybody, so I didn't know whether I could start or not. Okay, so thank you again for the introduction, Maria, Sara, and Julia. And today I'd like to share with you some concept, let's say, that we are trying to develop in the lab, which is about chromatin metabolism. And in particular, we do it in cancer, right? So, but before showing some results that we got in the last couple of years in the lab, I'd like to introduce you what chromatin metabolism is. So the concept of chromatin metabolism comes actually from my postdoctoral studies. I was working in Vienna, as Maria said, in the group of Stefan Kubicek, and there we were interested in understanding how epigenetic enzymes were regulating transcription in cancer cells, right? In particular, we were interested in an epigenetic enzyme, which is called BRD4. You can see represented here in purple, pink. So what BRD4 does, it binds to enhancer and promoters by recognizing acetylated histones, and then it brings there the transcriptional machinery, as you can see here in this square on the right side of the slide. So basically it interacts with transcription factors as well as with polymerase II, with mediator, and what it does at the end, it boosts transcription of specific oncogenes, like for example, MYC. At the time when I started my postdoc, the biology behind BRD4 activity was not that well known, so we decided to investigate it further, and we generated a report that was able to monitor the activity of BRD4, right? So with the reporter, we have done, with the reporter, we have done a genetic screening, and what we found was that the metabolic enzyme MTHFR1, which stands for methyltetrahydrofolate dehydrogenase 1, a very long and complex name, was interacting on chromatin together with BRD4. And this was very strange, right? Because this enzyme is supposed to be a cytoplasmic enzyme, and it does in the cytoplasm the reaction that, again, you can see here on the right part of the slide. So it converts, basically, it condenses first formate and tetrahydrofolate into 10,4-methyltetrahydrofolate, tetrahydrofolate, which is a precursor for the synthesis of purines. And then it does other two-step reaction to the 5,10-methylene tetrahydrofolate metabolite, which is needed both for thymidylate synthesis, so pyrimidine synthesis, or for the synthesis of SEBBM, which is the universal donor for methylation reaction. So it's required for histone methylation and DNA methylation, which then modulate transcription. So we didn't know what BRD4 and MTHFR1 were doing together, and therefore we investigated it a little bit more. And what we found was that they sit together at enhancers and promoters. As you can see here, this is a cheap analysis for MTHFR1 done with specific MTHFR1 antibody and BRD4 as well, together with H3K27. So H3K27 typically is a marker of actively transcribed chromatin, and it's present at promoter and enhancers of genes. And you see that BRD4 basically is there together with the H3K27 mark, and as well there is MTHFR1. But what they were there together for, right? So we discovered that not only MTHFR1 was sitting on chromatin together with BRD4, but actually other enzymes of the pyrimidine synthesis, and the pyrimidine synthesis were on chromatin. And this we have discovered by purifying the chromatin and doing mass spectrometry in order to identify what proteins were associated to chromatin in our cells. And they were there. So as we found basically the full pyrimidine and the full purine pathways on chromatin, we hypothesized that the enzyme were there to synthesize nucleotides directly on chromatin where they were needed. For example, a transcription start site, because the polymerase then it's able to incorporate, let's say, the nucleotides directly on the nascent RNA. And indeed, when we downregulated either MTHFR1 or BRD4 in our cells, we got very similar transcriptional changes, kind of indicating that those two proteins are involved in the transcriptional regulation of very similar transcriptional profiles. And this means, or this, let's say, led us to investigate whether this, this incorporation of nucleotides into the nascent mRNA was actually true. And this we have done by purifying the RNA and investigating whether, well, we did, sorry, by treating the cells with labeled formate and then purifying the mRNA and see whether in the mRNA we could find actually labeled nucleotide coming from the labeled formate. And indeed, this was the discovery that MTHFR1 together with the synthesis, with the purine synthesis and the pyrimidine synthesis was brought to chromatin by this binding with BRD4 to synthesize nucleotide and incorporate those nucleotides into the mRNA of target genes, as for example, MYC, as I showed you before. And importantly, we also demonstrated that the simultaneous inhibition of BRD4 and MTHFR1 together can actually be very beneficial when treating cancer, let's say, because it is very effective. So the combo inhibition is very effective, as you can see here, also in cancers that are not sensitive at all to either the treatment of BRD4 inhibition or MTHFR1 inhibition, which in this case was achieved with methotrexate. And this is what you are seeing here is a xenotransplant of this very aggressive lung cancer cell line, which is a 549, which is not sensitive to the single treatment. Okay, so with the discovery of MTHFR1 being on chromatin, we basically, switch from the canonical view that we have of metabolism to something that we like to call chromatin metabolism. So in textbooks, in biology textbooks, you will find that metabolisms typically occur in the mitochondria or the cytoplasm of the cell, right? So things are produced, metabolites are produced here outside the nucleus, and then they can reach the nucleus where they are needed, like for example, nucleotides for the transcription, just by diffusing and passing through the nucleopores, which are those big holes that there are present on the nuclear membrane, and allows, in theory, the diffusion of small molecules. What we are describing is instead that those nucleotides, specifically for the case of BRD4 and MTHFR1, were directly produced at the transcription start site so that the polymerase could rapidly use them to synthesize the RNA. So we wonder why, if there is this possibility of just diffusing from the cytoplasm to the nucleus, of why the cell would like to have the synthesis of nucleotide directly in loco. The reality is that cells are not full of water, they are full of proteins and cytoskeleton, and it's a very complex system and very crowded. And therefore, what we speculate is that, in this very crowded environment, actually diffusing, it's not something that is so much allowed, right? So synthesizing nucleotides directly at the transcription start site where they are needed to keep transcribing genes like, for example, MYC, which are important for the progression of the tumor, in this case, the proliferation of the tumor, it's going to be very beneficial for the tumor cells, right? Which also, make us see the other side of the coin. If we find a way to inhibit this production in loco, maybe we can arrest the transcription of those oncogenes and therefore also arrest the proliferation of the tumor. This led me to the idea of chromatin metabolism, and when I became a PI at the CRG, the project that I proposed was actually to investigate the role of chromatin metabolism in tumor progression. And this preliminary data, when I was, going from my postdoc to the PI position, I did some experiment to see what was the relevance, let's say, of chromatin metabolism in cancer. In particular, what you see here on the left is a chromatin proteomics experiment together with a nuclear metabolomics experiment. Here, the question that I was asking was, how many metabolic enzymes of the pentose phosphate pathway, the glycolysis pathway, or the folate pathway can I find on chromatin in cancer cells? And this is done I think if I remember correctly, with 3 or 4 different cancer cell lines. And the answer is that almost 60% of the enzymes of those pathways, and those pathways are not random, those pathways are the pathways that are involved in the so-called Warburg effect. So this is an effect in which the cancer cell decide to switch off, let's say, the respiration and upregulate the rate of glycolysis to kind of speed up proliferation. It's kind of hard to explain it in few words, but it's a very typical effect that we see in cancer cells. So 60% of the enzymes that are part of those pathways are actually present on chromatin, and basically nobody knows why, right? Some of them have non-nuclear localization, as you can see here in green, but again, the function in the nucleus of those enzymes is rarely described. And also we have performed nuclear metabolomics, and we saw that a bunch of metabolites that are actually associated, so either used or produced by those enzymes that are found on chromatin, were actually present in the nucleus in differential amounts when compared to the cytoplasmic counterpart, indicating that, you know, maybe activity of those enzymes is required in the nucleus to keep a different level of those metabolites in the nuclear environment, which would go then against the diffusion theory that I told you before. And this is what I saw. And therefore, with this idea in mind and this preliminary data, I wrote the ERC and I proposed in the ERC to then compare the chromatome of, so the proteomics at the chromatin level of healthy cells and cancer cells, to try to reconstruct on chromatin what are the metabolic pathways that we can find in healthy cells or those that we can find in cancer cells, and then check the, how different they are. And in particular, we want to ask 4 questions, which you can see here at the bottom, which metabolic enzymes are on chromatin cancer, how they can arrive to chromatin, right? So maybe cross-translational modification of different splicing lysoform or active nuclear transport, for example, may be some of the answers here. So where they localize on chromatin and why they are needed on chromatin, right? And here I just want to point out this beautiful logo that has been designed and realized by Rito, who is a PhD student in the lab, with very notable, let's say, artistic skills as well. So the first thing that we had to do was to then set up a Chromatin protocol that was reliable enough to be used in many different cell types, right? So I didn't go into detail, but basically what we are comparing now in the lab is the chromatin, the chromatin proteomics from 10 different tissues. For each tissue, we have at least 3 different cell lines in triplicates and so on and so forth. So this scales up a lot the number of samples that we have to prepare. So we had to have a protocol which was simple and reproducible. And I mean, the protocol is here just schematically represented. I'm not going into detail. If you would like to extract chromatin and do proteomics, you can just contact us. We can give you the protocol. But basically what I want to show you is that we don't see vinculin in the chromatin counterpart, in the chromatin part, which means that our protocol separates very well, leave the cytoskeleton, let's say, outside the nucleus. And also we enrich in H3 and we don't find FTX1. So FTX1 is a mitochondrial marker, and we were very happy to see that because, of course, as a lot of metabolic reaction are happening in the mitochondria, we wanted to be sure that we didn't have mitochondrial contamination in our chromatin preparation. And here you can see some immunofluorescence again, with DAPI, which marks the nucleus, and FTX1, which again marks mitochondria, and at the stage of the clean nuclei, which happens here, right? So from here, then we are just half protocol through, because still we have to break the nuclei and isolate chromatin. So at this stage, we already don't have mitochondrial contamination. And this is not just true for HeLa cells, it's true for basically all the different cells that we have tried, both normal cells or cancer cells. And here you have just some examples, and basically at the pure stage, which again is in the middle of the protocol, you don't see basically any LTS contamination, so any mitochondrial contamination. So what do we do with this protocol and with the information that we get from chromatin metabolism, right? So what we try to understand is how chromatin metabolism can influence actually the chromatin environment. What we know from my study as a postdoc is that, for example, the localization of chromatin of MTHFR1 controls, together with epigenetic readers, transcription. What Natalia will tell you in a bit is that MTHFR2 can control something else on chromatin, in particular centromer stability. What Laura is investigated, and she will probably show you something about it, is that there are metabolic enzymes that can control histone modification, and in particular, she's interested in histone lactylation. And then we have also found that metabolic enzymes can control the response against DNA damage, such as, for example, the PRDX1 enzyme or the IMPDH2 enzyme, as well as we are investigating the role of the chromatin localization of metabolic enzymes in cell cycle regulation. And today, just for sake of time, I will show you some results about the role of IMPDH2 in DNA damage repair. So here we focus in one specific cell line, which is in one specific tissue, which is breast. And in particular, the question that we were asking was, can we find metabolic enzymes that are associated to chromatin in very aggressive breast cancer cell types? And to answer this question, we compared the chromatin proteomics of 5 different breast cancer cell lines, which are T47D and MCS7. I forgot to write MCS7 here, which are hormone receptor positive, BT474, which are hormone receptor positive and HER2 positive, and SKB3, which are only HER2 positive, and the triple negative subtype, which is represented here by the MTA and B231. So all this positivity, let's say, represent different aggressiveness, right? So it's known in breast cancer that if cells are positive for hormone receptors, they are less aggressive. And if they are positive, for example, for the HER2 receptor, they are more aggressive than the estrogen and progesterone receptor positive one. And when they are triple negative, they are, the most aggressive one. And it's not only a matter of aggressiveness, and the aggressiveness can be explained, for example, by the fact that the estrogen and progesterone receptor are more epithelial cells, while the triple negative are more fibroblastic. They tend to invade, they tend to metastasize more. But also the problem relies on the fact that, the cells that are positive for some of those receptors, they need the activity of those receptors to proliferate and keep going. While, and therefore, if you block those receptors with, for example, hormone therapy in the case of estrogen and progesterone receptor, you can block the growth of those cells. While the triple negative breast cancer do not care. They don't rely on the activation of those receptors, and therefore we have no tools, we have no drugs to stop the proliferation of those cells. So the idea was to find metabolic vulnerability on chromatin and then use this information, let's say, to try to kill this specific aggressive breast cancer subtype. So we applied our chromatin protocol, chromatin proteomics protocol, and we found, without again entering into details, that there were a metabolic enzyme, which is called IMPDH2, that was significantly more abundant in the triple negative subtypes when compared to the hormone receptor positive or HER2 positive cell lines. And this you can see here in this representation, but also in the volcano plot of the mass spectrometry results here on the right. So IMPDH2, well, we have in the human cells, we have two IMPDH enzymes, so IMPDH1 and 2. And what those enzymes do is the conversion of IMP into XMP, which then is used for the synthesis of GTP. Actually, we can see this enzyme like a balanced needle, let's say, between ATP and GTP production, because it really, you know, determines the equilibrium between those two nucleotides. What we found was that not only IMPDH2 is more present on chromatin and triple negative breast cancer subtypes, but actually there are other enzymes, as you can see here, of the purine synthesis that are more abundant in these in the triple negative breast cancer subtypes when compared to the hormone receptor positive subtypes. And this is not just true for the 231. We can see, in fact, the presence of IMPDH2 here on chromatin, also in these other breast cancer, triple negative breast cancer subtypes, which are here represented in the Western blot with the pink text. But we don't basically find any IMPDH2 on chromatin in cell lines that are not from the triple negative breast cancer subtypes. And also, Laura in the lab developed MCS7 cells, which are hormone insensitive. So the MCS7 cell, as you can see here, as I told you before as well, they are they belong to the hormone receptor positive subtypes. But if you treat them, if you culture them without hormones, then they develop in culture the ability of surviving without hormone, and they become triple negative like, right? And so interesting, when they become triple negative like, they start accumulating IMPDH2 on chromatin. And we also know that the triple negative precancer cell lines are much more sensitive to the loss of IMPDH2, as you can see here. This is data from the Achilles dataset, which basically is a collection of essentiality based on CRISPR screening. And also we perform knockout for IMPDH2 in the lab for those two, for 231 and Cal51 triple negative breast cancer cell line. So you can see here that the knockout is very nicely working, and the cells basically are dying, as you can see here in the dashed red line at the bottom. And they survive only when we put in culture guanosine, meaning that the enzymatic activity of IMPDH2 is required. In parallel with a collaborator, we developed a chromatome experiment to identify metabolic enzymes that are preferentially localizing on chromatin during DNA damage. And what we found there, very interesting, was that IMPDH2 actually localized on chromatin during the recovery from the damage, as you can see here in the volcano plot. And in parallel, what was recently published in the Open Cell Consortium was that IMPDH2 is actually an interactor of topoisomerase 2A and PARP1, which are both very key, very important players in the DNA damage repair. And this was ringing a bell, let's say, in our head, because it's known that triple negative breast cancer often harbor mutations in DNA damage repair factors, or even they downregulate their expression to shut down the DNA damage repair and avoid apoptosis when DNA damage overaccumulates. Therefore, we believe that IMPDH2 on chromatin is actually signaling for DNA damage, kind of controlling DNA damage repair, especially in triple negative breast cancer, which relies on very few mechanisms to keep under control the damage accumulation. And in fact, if we inhibit IMPDH2 with MPA, MPA stands for mycophenolic acid and is a direct inhibitor of IMPDH2, we see an increase of DNA damage, which is quantified here with gamma H2X, which is a marker, let's say, of DNA damage. In addition, if we take our knockouts and we remove guanosine, we see that over time they accumulate replicative stress, so DNA damage due to stalling of the replication of the DNA, and also gamma H2X against which is a general marker of DNA damage, indicating that the activity, the metabolic activity of IMPDH2 is required to keep DNA damage under control. So basically, and this is my conclusion slide, what we know until now is that IMPDH2 localize on chromatin and maybe synthesize GTP on chromatin or keep the balance between ATP and GTP on chromatin to then somehow favor the DNA damage repair by interacting with topoisomerase 1 and PARP1. We have to decrypt a lot in the story yet, and basically how we are planning to do that is to identify novel chromatin partners of IMPDH2, which may play a role in DNA damage repair. We want to rescue the knockout with either the nuclear form or the cytoplasmic form of IMPDH2 to see whether the cells are able, differentially able to, overcome the DNA damage. And we want to understand whether there is a signaling role for the GTP synthesized directly in loco during DNA damage repair. And with that, I'd like to thank everybody in the lab, especially people that contributed to develop of the story, of the IMPDH2 story, which is Marta. She's a postdoc in the lab, Lorena, which is the lab manager and kind of multitasking person in the lab, and Carla, who's a bachelor student that joined a few months ago, but she's very active in all the immunofluorescence that we are doing with gamma H2X and LPA. And also, of course, the fundings, and you for listening. And if you have any question, I'm happy to answer.
Hi, Sara. I don't know if I can ask a question now or, yeah.
Sure, yeah, go ahead.
Yeah, sure.
Okay. Very nice results. I mean, I love it. Many congrats. And have you checked if this enzyme could also be involved in the pyrylation of PARP1?
We didn't. I mean, we just discovered this interaction with this open cell consortium. The publication was in Science, I think, last month. We didn't investigate it, but this is actually something that we want to investigate. And another thing is triple negative breast cancer. They are often treated with PARP inhibitors to actually kill them.
Yeah,
one of the idea that we have is really to try the combination of IMPDH2 inhibition and PARP inhibitors, in mice, for example, in which we do PDX with triple negative patients, and see whether we can actually kill the tumor more efficiently. That would be, yeah. Another strategy would be to do immunotherapy plus PARP plus IMPDH2. So try all this combination to see whether the addition of IMPDH2 inhibition can be more effective.
Okay, very nice. Thank you and congrats.
Thank you.
Is there some other question? Maybe.
Yeah, yes, I can ask one question.
Okay.
Yeah. Can I? Or?
Yes, go ahead, because I just copy another question that is in YouTube. Here is now in the chat, but you can ask first, Hannah.
Oh, thanks.
Sorry.
So yeah, very, very nice story, Sara. So I was just wondering, how about the genetic mutations? They are so prevalent only in the triple negative breast cancer and not in other ones, because you have seen only this you have seen only this interaction phenomena actually happening only in the triple negative. Do you know something about that?
No, I don't, but I don't think it's something that is happening just in the triple negative, right? Because I didn't have the time to go into detail, but for example, the chromatome experiments that we have done in collaboration with Joanna, the one that I showed where we find IMPDH2 on chromatin, this is not triple negative. This is U2OSL, so I think it's a general mechanism of kind of balancing And then we see also, this increase of IMPDH2 on chromatin at 24 hours after release of the DNA damage. So I think it's kind of a balance between repair of the damage and starting replication again, or starting cell cycle again, no? And then IMPDH2 is kind of there, kind of mediating this equilibrium, right? So I think it's happening in all the cell types, whatever, maybe also in normal cells, I don't know, right? But in the triple negative, they are more susceptible, let's say, to accumulation of damage because of mutations that they have in DNA damage repair. This could be one of the, remaining DNA damage surveillance protein that they rely on. And therefore, if you kill it, then they would overaccumulate mutation and die. But this is just a speculation. Big speculation. Yes, I can read the question. So thank you for the nice talk. Is there a nuclear localization signal in IMPDH2, or what is the mechanism to bring it to this compartment? It's a very nice question, actually. There's no strong nuclear localization signal. So it's not that we have done a deep analysis, but there are softwares online in which you can just upload the FASTA file of the protein and see whether there is some clear nuclear localization signal. There's not. So we would like to investigate whether some post-translational modifications are required for the transport. Also, with the proteomics experiment that we are doing, we can actually investigate whether IMPDH2 interact with importins or anyway proteins of the nucleopore complex to see whether it can actively be transported into the nucleus. Or another thing is that IMPDH2 size, which is around 55, if I'm not wrong, would even allow the diffusion, let's say, the spontaneous, let's say, passing of IMPDH2 through the nucleopores, because by size it would be allowed. But we don't know how IMPDH2 goes to the nucleus.
Okay, if there's no more questions, maybe we can proceed with the next talk. If there's any other questions, I guess we can read it from the comments in YouTube, also from Twitter, and answer after this presentation. So now we're gonna start with the elevator pitch talks. The first of our speakers is Natalia. Natalia studied biotechnology in Universitat Politécnica de Valencia, and she did a master in genomic medicine in University of Cambridge in the United Kingdom. There she worked on transcriptional networks in cancer. And after that, she joined the Sdelci lab in the Center for Genomic Regulation in Barcelona, where she's now a third-year PhD student. And now she's currently investigating the chromatin role of folate enzymes in cancer. The floor is yours, Natalia.
Okay, hi everyone, and thanks Julia for the nice introduction. So today what I want to share with you is this story coming from my PhD project, in which in the process of trying to characterize this non-canonical chromatin function of folate enzyme MTGF2 in cancer, we have discovered a novel role for this enzyme in mitosis. But first of all, why is MTGF2 important in cancer? Well, it was found out to be the most consistently metabolic gene overexpressed across cancer types. And since this publication, many other researchers have demonstrated the importance of this enzyme in driving several aspects of cancer development. And what is the biological function of this enzyme? Well, MTGF2 as well as MTGF1, they are both part of folate metabolism, which comprises a set of compartmentalized reactions taking place in the cytosol and in the mitochondria, and which is important for key biological functions, such as nucleotide synthesis, epigenetic maintenance, amino acid homeostasis, or redox defense. So in particular, we have MTGF2 here inside the mitochondria, where it catalyzes the conversion from methyl tetrahydrofolate into formaldehyde. But the exciting thing was to discover that this enzyme is not only present in the mitochondria, but also inside the nucleus in cancer cells. And it has also been shown to localize on chromatin. And because, as Sara very well explained, we are a lab doing chromatin metabolism, our main interest was to find out what is this folate enzyme doing on chromatin in cancer cells. So our first step here was to check whether MTHFR2 is recurrently found on chromatin or not. And to check this, we use an in-house subcellular fractionation protocol, which basically allows us to separate the cytosolic proteins from the chromatin proteins. And what we could see is that indeed MTHFR2 is present on chromatin in all these panel of cancer cell lines belonging to breast, lung, and colon cancer. So after this, we decided to characterize the MTHFR2 interactome, and we did so by pulling down the enzyme both in the cytosol and in the chromatin, and we sent these samples for mass spectrometry. So here I'm showing you a volcano plot of the top nuclear interactors, where we could see that many of them were involved in cell cycle regulation, which you can see here in green. And apart from this, we had a deeper look at this list of candidates, and we realized that actually many of them are important in mitosis, and they are related somehow with this interface between the centromeres and the kinetochores. So I will give you two quick examples. So for instance, we had P4A and TPR, which are both important for a spindle assembly regulation checkpoint, or a KMT5A and DNMT3B, which are methyl transferases involved in the methylation of the centromeric DNA. So because of this relationship between MTHFR2 and mitosis, we wanted to check, okay, what's happening in mitosis if we remove MTHFR2? So we generated in the lab some knockout cell lines using CRISPR-Cas9. And what we could see is that in both knockout clones, we observed a decrease in the percentage of mitotic cells, which can be measured by H3-phosphoserine staining. And apart from this, we also observed an increase in mitotic defects. So for example, here in both knockouts, you can see the presence of anaphase bridges, which means that these cells are having trouble in order to separate both sister chromatins during mitosis. And because we also knew that MTHFR2 was interacting with centromeric proteins, we decided to check the expression of the centromeres by RT-qPCR. And here what we could see is an exciting increase in the expression of these 4 centromeric regions, which was very interesting. And given the fact that these centromeric regions are marked with heterochromatin marks, we also had a look at those. And for example, here I'm showing you the H3K9 methylation 3, which is a marker of constitutive heterochromatin. And what we could see is that in both knockouts, there is a shift in the distribution of the intensity of this mark toward lower values. And this is very clear also from the images here. So the remaining question is why? What could be the contribution of MTHFR2 in all this mitotic function? So still we don't know, but what we hypothesized is that maybe MTHFR2 is needed in order to provide SAM in the environment, which is used for methylation reactions. And this can be important for heterochromatin maintenance and centromere stability. And we think so because one of the outputs of folate metabolism is methionine, which can then be converted to SAM. So of course, this is something we still need to validate, but with all the information that we have gathered up to now, this is the model that we are proposing. So what we think is happening is that when MTHFR2 is present on chromatin, it's interacting with centromeric proteins and it's providing SAM in the environment, which allows for a proper methylation and a normal expression of the centromeres. And in this condition, mitosis can proceed normally. However, if MTHFR2 is absent from chromatin, the levels of SAM are reduced, and this leads to decreased methylation and an aberrant increase of the expression of the centromeres. So in this case, what we have is problems with centromere stability, and this results in mitotic defects. And with this, I just want to thank all the members of the Sdelci lab, and especially to all the wonderful scientists that we have here presenting today. And well, with this, I'm open for any questions you may have.
Thank you very much, Natalia. I have actually a question. Maybe I missed it, but when you were talking about the results of the interactome and you found like all these hits that were involved in mitosis, did you find any hits related to other stages of the cell cycle?
With stages, you mean different phases?
Yes, different phases, exactly.
I mean, I guess we have a lot of signal of cell cycle regulation and G to M transition but nothing especially interest from G1 or S phase.
Okay, thank you. Do we have any other question? I can't see the YouTube, so I am not sure.
We have no questions for now, so if you want, we can go on, and if people want to post it, Natalia can answer later.
Okay, perfect. Then we will continue with our next speaker. Thank you, Natalia, for this very nice presentation. Our next speaker is Laura. She obtained her master's degree in Universitat Pompeu Fabra, working in the laboratory of Dr. Sandra Pairó in the Hospital del Mar. And she stayed there to pursue her PhD in the same laboratory. In 2016, the laboratory moved to Valdebrán Institute of Oncology, where she defended her PhD in 2018. In 2019, she joined Dr. Sara Sdelci laboratory, and her project is mainly based on characterizing the newly discovered histone lactation mark in the context of breast cancer. The floor is yours, Laura.
Thank you very much. Can you see it? Can you see my presentation?
Yes.
Okay, thank you. So thank you, Julia, for the nice presentation. So yeah, today I'm going to talk about the role of this newly discovered histone activation mark in the context of breast cancer. So in normal conditions, in normal cells and in the presence of oxygen, glucose is metabolized into pyruvate through this process called glycolysis. And then this pyruvate enters to the mitochondria to produce this CO2 in order to fuel the oxidative phosphorylation and generate 36 molecules of ATP for each molecule of glucose. When there is no oxygen available, there is a switch from the oxidative phosphorylation to glycolysis, and therefore cells use this glucose to produce mainly lactate through this anaerobic glycolysis process, but is much less efficient, producing only 2 molecules of ATP for each molecule of glucose. In the context of cancer, however, there is a very deep and complex metabolic rewiring in order to fuel the proliferation capacities of these cancer cells and therefore allow them to metastasize. This process is called the Warburg effect or aerobic glycolysis, which is mainly based on the fact that these cells, independently of the presence of oxygens, metabolize glucose into mainly into lactate, with the production in this case of 4 molecules of ATP for each molecule of glucose. This Warburg effect is particularly important for these cells because they are able to metabolize 10-fold more glucose to lactate into a given time as compared to normal cells, so therefore they are more prone to proliferate and to metastasize and invade. During the last century, lactate has been considered as a waste product of this of this Warburg effect. However, recent evidences suggested that this metabolite can be actually in many cellular contexts. So in this regard, in 2019, it was described this new histone mark, which is called histone lactylation. And in this sense, the authors described a new role for the Warburg effect, which is to induce the production of lactate to fuel this lactyl-CoA that will be therefore used for the lactylation of histones. And in this sense, this histone lactylation is mainly involved in the expression of genes. And in this case, the authors studied the expression of homeostatic genes that are required for macrophage polarization. However, we think that this histone lactylation may play a crucial role in the context of breast cancer, and in particular, we're interested in characterize this histone lactylation in breast cancer. To do so, we will try to decipher how does this histone lactylation happens and what is particular the role. As Sara already mentioned, the breast cancer can be classified in different subtypes depending on the expression of estrogen receptor, progesterone receptor, or human epidermal growth factor receptor 2. So the presence of those receptors actually generally accounts for the classification of breast cancer patients into good or bad prognosis. Therefore, those patients that express this hormone receptor they benefit from hormonal therapy, which have shown very good results in clinics, let's say, and they have better prognosis. The problem comes when these cells do not express any of the receptors, therefore they grow independently of the activation of the receptors. And in this case, there are no current therapy available yet. So therefore, those patients are in the worst prognosis among all the triple negative, among all the breast cancer subtypes. So typically, cells that are positive for this hormone receptor have this kind of epithelial phenotype. So the cells are now able to migrate and invade, or less able to migrate and invade. But for example, here you can see that these MB231 cells, which are triple negative breast cancer cell lines, they have a more mesenchymal phenotype, which will allow them to invade, and therefore they are much more aggressive. So in order to study which are the differences between histone lactylation between different breast cancer cell lines, we in vitro generated these hormone-insensitive cell lines, which should resemble this triple negative breast cancer phenotype. And we started from hormone receptor positive or sensitive cell lines in order to have an isogenic control. So to do so, we use these T47D or MCF7 cells, and we culture them at least during 3 weeks in the absence of hormones in the medium. So they theoretically will become hormone insensitive, and they will be triple negative breast cancer-like phenotype, let's say. So as you can see here, when we treat these T47D cell lines, we were not able to achieve the results we were expecting. So the cells stop growing and they become kind of senescent. However, in the case of MCF7, after 4 weeks in culture without hormones, they become insensitive, they start proliferate, and as you can see here, very interestingly, they completely change the phenotype. And this phenotype resembles the one from the triple negative breast cancer cell line that is very well established, which is the NDA231. In order to characterize further this newly generated cell line, this hormone insensitive, we treat them with the typical hormonal therapy, which is tamoxifen. And as you can see here, these cells become much more resistant. So this is corroborating the idea that we actually generated a hormone insensitive cell line in vitro. So given the fact that, as I told you before at the beginning, during the Warburg effect, when the cancer cells become much more aggressive, there is a huge production of lactate. We thought that most probably this lactate will induce histone lactylation, and this would be related to aggressiveness. However, when we perform the Western blot using a panel of different cell lines, these 3 cell lines here are triple negative breast cancer established cell lines, and we also use the newly generated in vitro. You can see that when we check different lactylation marks, those triple negatives show less levels of lactylation. And also very interesting, if you have a look at the T47 hormone deprived that do not proliferate, so they become senescent, they have higher levels of histone lactylation as compared with the parental. And in the case of the MCF7, which resembles the triple negative phenotype, here you can see that when these cells are hormone deprived, the levels of lactylation decrease and is much more similar to the triple negative breast cancer cell lines. So this allows us to conclude or to think that most probably histone lactylation is somehow related with lower aggressiveness, which is the contrary that what we thought at the beginning, but still I think it's very interesting. So after that, we wanted to know if it's indeed the lactate could be the source or the substrate of this histone lactylation. So for that, we took the T47D as high, MCF7 as medium, and then the NDA231 as a low lactylation cell lines to compare the lactate levels. As you can see here, extracellularly, lactate concentration tend to decrease when the histone lactylation increases. But importantly, and more interestingly, when we check at the intracellular lactate levels, there is an increase in the lactate intracellularly when there is an increase in lactylation. So therefore, suggesting that this much probably the lactate is the substrate of this lactylation. So the metabolic network required for the generation of this histone lactylation has not been deciphered yet. So in order to try to find which could be the metabolic enzyme responsible for this histone lactylation, we set up a functional genomic approach coupled to flow cytometry. to systemically identify these enzymes, metabolic enzymes, right? So what we did was to use CRISPR-Cas9 metabolic guide library to infect NCF7 cells. And then we tried to find which are the enzymes whose loss would cause either an increase or decrease in histone activation. Cells will be stained with for immunofluorescence and fact sorted according to their levels of this histone activation. In particular, cells showing the brightest and the dimmest signal will be sorted. Therefore, the genomic DNA will be extracted and deep sequencing to find which are these metabolic enzymes. We're still analyzing this data, but we hope it will shed light on this, the metabolic enzymes required for this new described histone activation. So as a conclusion, we have shown that triple-negative breast cancer cells have low levels of histone activation. Therefore, histone activation might be related with lower aggressiveness. And we have seen that lactate might be the source of this new identified histone mark. However, still there are a lot of questions to answer. So for example, where does this lactation happen in the context of breast cancers? For that, we have done chip sequencing, which is actually the substrate, the direct substrate. What is the consequence of this histone activation, and when does it happen? So with that, I would like to thank to all the Sdelci lab for their feedback and their support, and of course to all of you for attention. If you have questions, I will be happy to take them.
Do we have any question in the YouTube?
Not at the moment.
Okay.
I have a question.
Okay,
Go ahead, Anna.
Thank you. Thank you, Lara. That was very, very beautiful. So I have two questions actually. So first question is like, have you looked at the human samples to see actually what is happening to your lactation histone? So do you see, and especially I guess in the breast cancer patients?
No, I haven't. But this is, I mean, this is something you mean in the protein atlas, for example, or something like this. Yes, we have checked for some enzymes, particular enzymes that at the beginning we thought could be related to that, but we should have a deeper look on that for sure.
Okay, and the other question that I had was actually related to have you, or you may be thinking maybe treating the cell. So basically what happens after the treatment, no? Because if you would like, I don't know, perform some chemotherapy or, you know, some chemotoxic drugs on your cells. So what will happen actually? So
you mean upon treatment with tamoxifen, for example?
For example, yeah, which is basically the standard care for those patients. Yes.
Yes, this is something at the beginning we wanted to do. Actually, we wanted to produce these hormone-insensitive cell lines, and after that treat them with tamoxifen, so they will become tamoxifen resistant, like super resistant, right? This is something I didn't do at the end, so we finally end up with this hormone insensitive. We were kind of happy because it took a long, but for sure for the experiments that we're performing now, for example, I'm performing chip sequencing, I'm performing RNA sequencing and all this stuff. It would be very, very nice to see what's happening with hormone, you know, insensitive and after tamoxifen treatment for sure. And see if the cells, you know, well, I know that the cells respond differently, but what is the consequence, right, of this resistance? Yeah, for sure. Thank you for the question, Anna.
Okay, maybe I can jump in because now is, if there are no more questions, it's Julia's turn for a 5-minute pitch talk. So Julia was presented already by Sara at the beginning, so I will try not to repeat myself a lot. So she's a shared PhD student between my lab and the lab of Anna Janic at the UPF, and she did I think she has been interested in science since she was born, more or less, and she has tried to, to be involved in laboratory experiences. She was 15 or 16, which is, I think it's amazing, which shows how passionate she is about science, right? And this is also reflected in the many awards that she won already. I think for sure she won more awards than I did. Apart from the fellowships, I mean, she was awarded also the Youth and Science Scholarship from La Pedrera Foundation. She won the Regional Chemistry Olympiads in the bronze medal also for the Regional Biology Olympiads. They're crazy about biomedicine scholarship. So, she's really into science and into research, and both me and Anna are very happy to have her as a PhD student in the lab. So yeah, Julia, the floor is all yours.
Thank you, Sara, for this very nice introduction. I actually loved science since I was very, very little, but it's thanks to like all the people who has make it like this beautiful to me, such as you. So thanks a lot. Well, thank you very much for being here today in this webinar. I'm gonna give you a bit more conceptual presentation today, since I just finished my first year, so everything will be more theoretical instead of results. But basically, I'm going to give you some insight of my PhD project. I am focused on understanding which are the metabolic pathways that are essential in specific type of lung cancer, which are known to be a bit more aggressive than the other. Well, actually pretty much aggressive than the other. And the first thing I want to point out is why we want to study lung cancer. And the reason why is because if we take into account the data provided by the World Health Organization, we will find out that lung cancer is actually the deadliest cancer. It counted per 1.8 million deaths in 2020, and it's the deadliest one with difference from the others. But it's not only that it's the deadliest cancer, but it's also one of the most highly diagnosed cancers, with 2.21 million new cases in 2020. And I think that the most important highlight actually from this slide is the fact that if we take a look into breast cancer type, we would see that even though the amount of cases is even higher than lung, the number of deaths is less than half than the one caused by lung cancer. So we still miss a lot of information in order to find new treatments, etc., to provide better cures for this type of cancer. In my project, I'm focused on specific type of cancer, which covers 80 to 85%, which is non-small cell lung cancer. This is like phenotypically distinction between small and non-small cell lung cancer. And more specifically, I focus in a 15% of these non-small cell lung cancers that contain a co-mutation in KRAS and TP53. And the reason why we focus on this is because these double mutant, this co-occurrence is the one that leads to this more aggressive phenotype that produces worse survival rates for the people who has these specific mutations in the genome of the tumors that they have. So the thing is that even though KRAS and TP53 are widely studied, there's not a lot of understanding of the molecular mechanisms that are caused by this double mutant, and this is what we focus on. And of course, if we take a look into KRAS and TP53 in the literature, these are two of the most mutated genes in cancer. And most recently, they have been related with the metabolism. This is actually a very hot topic now, and it's what my PhD project is actually based. TP53 and KRAS are known to be involved in metabolic rewiring changes in the cancer metabolism. And more specifically, if we take a look to what is available about TP53, we will find out that TP53 is involved in energetic paths such as oxidative phosphorylation, lipid metabolism, glucose metabolism. Also glutaminolysis might be also related to this oxidative phosphorylation, and other paths such as the novoserine synthesis. KRAS on its own, it's also related to other pathways such as autophagy and macropinocytosis, like introducing and degrading compounds inside the cell. But it also shares other pathways together with TP53. But the important point of this is that even though we have a lot of information of these genes separately, how do they regulate the metabolism, there's very, very few information about what happens when the two mutations co-occur. And that's what we aim to discover, what we aim to find. We hypothesize that these co-mutations, these co-occurring mutations, deal and produce specific metabolic rewiring that we could actually target. So we want to know what is essential metabolic-wise for this specific type of double mutant non-small cell lung cancer. To do so, my project is basically divided into processes. The first is identifying what is essential for these type of non-small cell lung cancers. The second one, it's based more in the characterization, like knowing how these metabolic essentialities produce, like what do they produce in this cell that makes it essential. And to do so, I am actually using a strategy. It's very similar to what Laura mentioned in the end of her talk. I am using different types of non-small cell lung cancer cell lines, and I am infecting them with a library of guide RNAs, which will allow me to knock out different metabolic genes inside these cells one by one. And so we can see in a big picture what happens with the cells when each of these metabolic genes is knocked out. An important part of this project is that we aim to achieve different perspectives, different approaches that would include things in vivo, such as 2D and 3D. The 3D in this case will add to the whole picture the structural architecture and cell-cell interactions that it's actually missing in the common used 2D cell culture. And also we want to include the in vivo system in order to see also what can we translate from the dish, from the plate to the whole organism. We want to differentiate as well between the immune competent and the immune deficient systems to see if there's actually an immune metabolic interaction that might be relevant in order to understand how these metabolic essentialities work for this double mutant KRAS and TP53. Once we got this information and we put together all this data, but this is going to take a bit of time still, but we will try to knock down all these genes and then see how the tumorigenicity by checking the migration and invasion capabilities, proliferation, sphere information changes when these metabolic genes are knocked down in this double mutant KRAS and TP53. And after that, we will want to know, like transcriptomic wise and metabolomic wise, what happens inside the cell when these knockdowns are also produced. But now I'm going to show you just like one last slide about the summary of the results that we obtained for the first screening that we performed in this 2D system, the first preliminary data that we obtained that actually was very promising. And we found out that there are like 3 main paths that might be actually more essential for this TP53 KRAS double mutant, which are 2 involved in post-translational modifications, such as N-glycan synthesis and GPA anchor proteins, meaning N-glycosylation, changing the N-terminal of the protein, and some modification that allows some proteins to stack into membranes. And another very interesting part was the fact that it seems that the purine synthesis might be actually very relevant for this KRAS TP53 model. This purine synthesis basically is what it's going to lead to guanosine and adenosine production in the end. So this opens a lot of doors, let's say. We are now trying to validate this data in the specific cell line that we use for this, but we would like also to know if these results correspond as well to the ones that we're going to obtain in further screenings that we are currently developing in the lab. With this, I would just like to thank all the people involved in both labs, of course, Sara and Anna, my supervisors, and all the people that have been evaluating my project as well. And thank you, all of you, for your attention. And now if you have any question now or later, I would be very pleased to take them. Thank you very much. I don't see any question here. I don't know if in YouTube, if not, we can continue.
Yeah, I would say that we can continue. And then if there are questions either at the end or just reach out to Julia, she'll be happy to answer.
Exactly. Okay, so now we have the last elevator pitch of today. The last speaker is Edna. She graduated from veterinary medicine at Universitat Autònoma de Barcelona. After that, she studied a master in translational biomedicine, and she joined there the group of cancer stem cells in Vall d'Hebron. After that, she started her PhD under the supervision of Anna Yannick here as well, also in this webinar since 2018, and now she's a fourth-year PhD student studying the tumor suppressor mechanisms of TP53. The floor is yours, Edna.
We can't hear you, Edna. I think you're mute.
You're still muted.
Now?
Now, perfect.
Okay, so let me check. I have so many things open that I cannot. 1 second. Okay. Okay, now I can see. Share. Can you see the presentation?
Yeah.
Okay. So today I'm going to talk about this weirdly named protein, RNF144B, which is quite unknown, but it plays a role in the signaling pathway of P53, which is a very well-known tumor suppressor. In our group, we focus on the study of P53 tumor suppressor because it's a critical tumor suppressor, and this is stated by the fact that 50% of all cancers of all types have a P53 mutation. Moreover, if you have a germinal mutation on this gene, you will develop Li-Fraumeni syndrome, which is a rare disease that predisposes to early onset of cancer. And finally, if you knock out P53 in a mice model, 100% of all mice will develop tumors. It is known that P53 has an important role in regulating many cellular protective pathways, but in my project, I focus on DNA repair and genomic stability regulated by P53. So upon perceiving DNA damage, P53 is stabilized and it can bind there to the DNA and acts as a transcription factor. It's able to activate around 300 genes. And here I'm just pointing out genes that are transcriptionally activated by P53 and that take a role on the DNA repair. So you can see that there are many proteins that are involved in several different DNA repair pathways. However, there is still a subset of proteins that are involved in DNA repair and are activated by P53, but we still don't know exactly in which stage or process do they participate. And specifically, my project focuses on this protein called RNF144B. And why I'm interested in this protein? Well, it was shown in a previous study from Anna Yannick, our supervisor, that if you knock down RNF144B in a MYC-driven lymphoma model, so basically she injected hematopoietic stem cells that carried the MYC oncogene, and you can see in the black line that eventually all control mice due to this MYC expression eventually die because of the lymphoma formation. However, if you concomitantly knock down either P53 or RNF144B, these mice die much quicker. They die at 50 days of age. So this gene already showed a promising tumor suppressor role in this lymphoma model. Let's get to know a bit better this protein. So this is a G3 ubiquitin ligase. What it does is it ubiquitinates some unknown target proteins, and therefore this tag of ubiquitin sends the proteins for degradation. What we also know is that it is closely working in this P53 signaling network.
because it has been described to being activated by P53, but also by its siblings P73 and P63 in an autoregulatory feedback loop. Moreover, it has shown to degrade P21 and PAX, which are critical for cell cycle, apoptosis, differentiation. And finally, it has been shown that if you knock down this protein, you will have more DNA damage. However, the mechanism is still not well understood. So my project focuses on the role of RNF144B in tumor suppression, not only in lymphoma, but translating it to other solid cancers. And specifically, I'm working in lung cancer. I do it in a similar way to Julia, but without the screening. So basically, I infect some human and mouse cancer or healthy cell lines with lentivirals carrying the CRISPR components to knock down RNF or P53. And then I perform in vitro 2D studies or in vivo studies, either in a subcutaneous manner or orthotopically into the lung. And I study different cellular functions that are key for the P53 pathway, like proliferation, DNA repair, cell cycle, or apoptosis. And finally, as I am working with ubiquitin ligase, we have also performed a proteomics study to try to find some targets for degradation of this protein. And now I'm just going to show some preliminary results that are nice to share with you. So one striking result that we saw is that these are chromosomal spreads, metaphase spreads, and the regular number of chromosomes in mice cells are around 40. However, we can see that when you knock down either P53 or RNF144B, these cells gain, they have a doubling of chromosomes. And the cells present around 80 chromosomes. So it seems that RNF is preserving the genomic stability of these cells. Moreover, we irradiated our cells. In this case, I'm working with mouse embryonic fibroblasts. I irradiated these cells and I measured the gamma H2X, which is a marker of double strand break. And what I observed is that 1 hour after the irradiation treatment, the P53 knockdown cells and also the RNF144B knockdown cells presented higher intensity of this signal, so higher DNA damage. Regarding the in vivo studies, I also checked the capacity of the knockdown cells to form tumors into mice. So these MEFs barely grow, these fibroblasts barely grow in vivo, subcutaneously. However, when we knock down P53 or RNF144B, we can see that tumors grew much quicker and bigger in these cases. And finally, I took a lung cancer model, human in this case, which is A549. And what I do is I inject 200,000 cells orthotopically into the left lung. And I wait 6 weeks for the tumor to grow. And finally, I collect the tissue and I can study histologically the tumor there. So here you can see, here what I do is I measure the area of the tumor between the control cells. And in this case, I have full knockout. So these cells, these A549s, don't express at all RNF or P53. And we can see that the area of the tumor is bigger in the RNF144B knockouts and the P53 versus the control. So these proteins seem to have a role in tumor suppression, not only in lymphoma, but also in other cellular contexts. And now just to finish, I thank my team, my supervisor, and also any other lab group that has helped me with the regions and everything. And now if you have any questions.
I do have a couple of questions. Very interesting. It's very nice to see how the story evolves. And
yes, it's slowly, but it's evolving.
It's not that slowly. It's a lot of work. So I have a question about this number of chromosomes, right? So yes. Those are knockout cells?
They are knocked down, these cells. They are the MEFs, the same MEFs. Yes.
Okay, because it's kind of striking that you have either 40 or 80, right? So you have really two clouds of population, which means, I don't know, that the cells replicate without dividing at the end, right? So they replicate and they probably do not undergo mitosis, or they do mitosis, but they don't do the cytokinesis, and they keep the two nuclei together.
So with these spreads, I'm curious about this mechanism because I don't completely understand it yet. But what we do is we treat it with colsemate, which is a destabilizer of the microtubule. Yes. So in theory, the cells are they cannot divide, so the chromosomes are packed, and they are in the condensed shape, but they are not entering mitosis. And in that moment, I proceed for performing the metaphase spread, and I see this difference. So
could it be due to the levels, meaning that in some cells you have more RNF, so more similar to the wild type situation, and then, because of the knockdown, and then these keep somehow the ability of whatever, surveilling the genome and segregate the daughter, let's say, genomes correctly. And in cases in which you have a strong downregulation of RNF, you lose this capability, and therefore you have this, aberrant cell cycle retaining the two nuclei inside the cells.
I haven't put here because it's a short presentation and also it's not solid, but we have hints that, for example, there is an increased G2 phase that I'm trying to study further, because maybe the lack of RNF144B is affecting the entrance or the correct mitosis. We are also trying to study with immunofluorescence possible aberrations in the mitosis like Natalia does. And also it has an effect in apoptosis. We have seen that these cells survive a bit better to several treatments. So maybe because if you have chromosomal aberrations, the cells usually through P53, they go for apoptosis. So maybe if both pathways are affected, these cells survive and are still growing even if they are aberrant for their chromosomal content.
It comes to my mind because we have the Fuji cells in the lab, right? So with the Fuji cells, you can actually label cell cycle, right? It would be very interesting to see tracking cell by cell in your RNF downregulated population, whether you have cells that actually go back in the cell cycle. So they go into G2 and then they cannot progress into M. This shouldn't happen normally, right? Because the mitotic checkpoint should avoid this, but if they have also defect in sensing apoptosis, they may go back. They may be able to go back, and therefore that's why you see this duplication of the full genome rather than having, different copies of different numbers of chromosomes. Because due to instability, I would have expected to have like for 40 to 80, but all the numbers, right? Like spread.
Yeah, and for P53, yes, it's interesting. And also for P53 knockdown, so it's not only my results, that there is a paper published that they also show this trend, either 40 or around 80, but not spread. So like
this could P53, it could be a mechanism of I think someone is saying something.
Yeah, it's me, Natalia.
Yes.
No, sorry. I just wanted to say that whole genome doubling is something that happens with TP53 loss. So I think this is what up this 40-80 thing, because the whole genome is just doubling.
Yes, it's very interesting.
Very cool.
It's very cool. And my other question would be, because you saw that if you irradiate the cells, you have much more DNA damage in the TP53 and RNF knockout or downregulated cells. I don't remember now. But what about recovery?
Yeah, I need to perform. So I'm eager to do that for like several time points, and also more long term, like 1 day or 2, to see the remaining DNA damage that could be there.
Okay, that's cool. Very cool. Congratulations.
Thank you.
Well, I can stay here. I don't know whether there are more questions in the YouTube channel.
Not for the moment. We had one on the Q&A in the Zoom, but Julia answered that already. It was for Julia. So if you want, we can go ahead to the roundtable.
Yes, sure. So for the roundtable, I think we all switch on our cameras. And yeah, I'd like also to introduce Anna that is here with us. So Anna is also a group leader at the UPF, and she's the master expert on P53 biology in cancer. And before coming to the UPF as a group leader, she was a postdoctoral investigator in Australia, in Melbourne, and the Walter and Eliza Hall Institute. And she has been at the Marie Curie postdoctoral fellowship, and she has published beautiful papers. The last one is postdoctoral fellowship in natural medicine, which is basically the basis for funding her lab, let's say, in Barcelona. And this is what, for example, Edna took over, and she's further studying and developing based on those discoveries. So Anna, thank you very much for being here. And yeah, it's very strange to have a round table on a screen. But yeah, let's try to do it our best. So the round table was supposed to be like very spontaneous. I mean, it's going to be very spontaneous because we didn't prepare anything, right? So I don't know whether Julia, who's the other moderator, have a question that want to ask to all of us, or to the postdoc, could be Laura here, or to me and Anna, or to your peers, Edna and Natalia. Or otherwise, I don't know what, if some of you may have a question, or someone from the audience have a question that wants to ask, and then we can start the discussion. I don't know, Julia, if you have any question that you didn't dare to ask during the last year that you've been in the lab.
The truth is that for me, most of it was pretty clear. I'm sorry because I hear me double now. I'm sorry, Edna, because we are in the same room with Edna. Can it be something? I want to ask a question in general. Can it be something more trajectory-wise, or can it be something?
Sure. Yes, sure.
Okay. So I would like to ask to all of you, which did you think was the most challenging moment of your PhD? Since I am now, like, I have started already, but I still have a long way to go. Maybe someone can tell me, what do you think I should be ready for, and how to overcome it?
Okay. Who wants to answer?
Maybe I can answer. Okay. I mean, I have to say that I'm this kind of person that I really enjoyed my PhD. So I was very, very lucky. However, of course, there are a lot of moments where you struggle with a lot of things. I would say that maybe the most probably difficult moment, because it not only depends on you, but also on other persons. Yeah. It was the moment that I had to publish my results, right? So normally you feel that you have, so this is something that is not only happening in your PhD, so actually it's happening to me right now. But normally you are so very happy, you are, enjoying the research, enjoying everything, and suddenly you have to write everything in a single manuscript and send it, and other people, criticize it. Sometimes it's good feedback, but sometimes it's not. It just, I don't know, your work is not good enough or whatever. So this was kind of hard for me to try to understand what's going on actually, and try to figure out how to improve everything. But it was complicated, but it was also very enriching, because you learn a lot, and then you have another kind of thinking, right? You have a more critical thinking on your experiments. So I think this is a very good thing that that happens to you, but at the moment is hard, let's say. Yeah, I would say it was that moment.
Well, I can also add something. Yeah, I completely agree with Laura that publishing papers is not the easiest moments through the career. But I have another story, which I think is for every student and a postdoc, and even for us now, is the most important, is to challenge our results, right? To revise them and to, you know, look at them from a different angles. And because during my PhD, what happened is that I had like beautiful phenotypes. I had these fruit flies, which were called Drosophila, and they had tumors. And then I downregulated, well, I knock out some genes that I thought I found by transcription profiles that are so important for the growing of these tumors. And I knocked them out, and then the tumors didn't grow. And then the flies, so this happened like in the year between second and third year of my PhD. And then in the fourth year, when we wrote the paper and everything, it's about to send it. And then those flies started to get tumors again. And then I was like, okay, because you have these stalks, right? And then, you know, you were thinking, oh yeah, but these flies, they didn't, you know, they didn't develop tumors. They were living, flying, and then they start to die. And I was thinking, oh my goodness, what happened this? And then, you know, it was like the moment that was for me very difficult first to explain to the supervisor, you know, what happened. And then basically, you know, that what basically we found that is like shrinking the tumor in one of the stalks. It didn't happen again. So this was very, for me, like difficult to approach, you know, this conversation. But yeah, I went, and then it was really actually made it much even better, because, you know, we could actually found like genetically what happened to these stalks, and it was just like technical, like some Drosophila genetics phenomenon that sometimes can happen, going back, doing another, so making another double mutants, and we could again confirm the phenotype. But I think each of us shouldn't be afraid of, you know, like their results, standing there and, you know, discussing and also revising, you know, ourselves and criticizing, asking the questions, which I think is, it is very important. So, but yeah, it was challenging to see that.
Yeah, actually, I very much agree with Anna. I mean, sometimes that the most catastrophic results, right? That you think, okay, this is not working at all. This is disaster. All the work I've done till now is not gonna, you know, sometimes they are the most interesting ones at the end. So sometimes they are the ones that change everything and everything fits even better or whatever. So yeah, this happened to me as well. So we were expecting some results, and at the end we obtained a completely, you know, the opposite effect. And I was like, oh, come on, this is not possible. And at the end, this was the main point of the paper, so the strong point of the paper. So it was nice. But yeah, I agree that this is also a very complicated moment.
Yeah.
I think Natalia wanted to say something, right?
Okay, I was mute. No, well, I'm in the same journey as Julia, so I'm not at the level of that experience yet as like postdocs or of course PIs, but from myself, like the worst thing is dealing with frustration, because I was used to, as a student, to have this thing that if you work harder, you will get better results. But in real life, experiments don't work like this. Like even if you do super hard work, like extra hours, whatever, doesn't mean you're gonna get the result that you want. And then coping with, like, I don't know, like these experiments are not working, and you even like sometimes don't trust yourself or don't trust what like the story is doing or where it's going. And yeah, all this frustration, it's difficult. And I think it's the main thing you learn in the PhD, how to deal with these like negative results that sometimes I agree with Laura. I hope that everything ends well at the end, but yeah, the journey is complicated.
Yeah. I think, I mean, you are all right? I experienced everything of it. I remember several times when I was a PhD, I was having nightmares about short-term things not working or things like these cells dying or whatever, you know, in the middle of the experiment, because those were things that were really happening during the day, you know? And I was so frustrated, I was even dreaming about it. And it was really terrible. So, and also I think there is this, time limitation, which is very marked in a way, right? You have to finish it in 3-4 years, that's it. And there is a learning process which takes almost 2 years, in which you are growing, in which, I mean, the first year is basically understanding where all the reagents are in the lab and how to mix them without making an explosion, basically. And the second one is when you start understanding what your project is about. And then finally, the third and the fourth one is when you are, fully capable of doing whatever you want, even thinking about experiments and moving forward the project, is when you have to leave. So the frustration comes also on top of it, right? Because the moment in which you will publish your story, for example, Julia, right now, Natalia is the tip of the iceberg, right? So then if you, from your stories, more stories will born, and then there is also this moment in which it was very frustrating for me. For example, when I left the PhD, it was like, okay, but I have those stories, they are all at the beginning, so what do I do with them? Who is gonna take care of them? And especially in the lab where I was a very small lab, I knew that nobody basically was going to take care of them. So I just left them there, and it was so sad because those were results that we never published at the end, right? And it was frustrating. But on the other side of the coin, I think that, because I did 2 PhDs. One I didn't finish, one I started and didn't finish, and the other one I finished. Thanks God. and I think that, for me also it was complicated. For example, during the first PhD, the frustration was not coming from the experiment, it was coming, and this connects with what Anna was saying, right? Having this open relationship with the supervisor and not being afraid of saying whatever comes to your mind and whatever about the experiments, or even if you did mistakes and so on, because the frustration in my first PhD experience came from the fact that I didn't have this relationship with my supervisor, right? So we were actually very much in conflict, and this was one of the reasons why then I decided to stop that PhD and start another one. And that was complicated, right? So you cannot really manage all the frustration and all the failures, right? If you don't have a person supervising you in which you can rely and, and can accompany you in the journey, right? And then at the end, I think this is fundamental. So Julia, what's the biggest difficulty for you so far?
I think that it's learning a lot of things from scratch. Like there's a lot of techniques that I've never done, or type of experiments that I could never imagine before coming here probably. And now it's like looking a lot into literature, trying to figure out which is the best way. And it has become challenging, but it has been it has been amazing as well, because somehow I end up being able to do things I didn't even know I could do. So I'm happy that, even though sometimes it comes with what you say, like it doesn't come out well the first time, so you have to try another one, but I think it's very nice.
I have to say that, Julia, you are doing a very great job in this sense, because I think you're going in very, deep detail on the techniques and what you are doing, and trying to understand very well actually what you are doing, that sometimes is not, the norm. So I think you are doing a very good job in this sense, and it will be very, very, very useful in the future, because you are facing this, but this will happen as well. I faced this as well when I started the postdoc, right? I changed the field kind of, and there are a lot of things that I never heard about and no idea. So if you now are used to do it, it will be much easier in the future. So I think that everything we do right now that we're struggling will be very useful in another moment of our career, for sure.
Thanks a lot, Laura. Yes, I actually think so too. I feel more ready in life. Even how I deal with things in life has changed due to this, I think.
Yes, and I think that this is mainly also due to what Natalia was saying, right? That you have to deal with frustration. That was normally when you arrive at this stage of your career or your life or whatever, you are not very used to that. And this is very true. When you are studying, the most you study, the better grades you will have, most probably. But this is not applying to science or to research, right? So it's, yes, it's kind of a, you know, I don't know, this is something that you can apply to life as well. Yes. And also now you have the frustration, and sometimes you feel you are not doing well, and then it comes, or maybe it's the same, this kind of imposter syndrome it's always in your head, right? Saying like, you are not able, you are not good, you are not whatever. And then you have to hard work to demonstrate not only the others, but to yourself, which is even more important, that you are actually able to do it, or kind of able to do it, and that you are not stupid, right? Because, you know, the other day we were in this kind of also roundtable with other, in this case, were mothers in science. And this case, we were talking about that, and someone said, okay, but we are all saying that we have to hard work, you know, to persevere. I think it's so, I don't know, it's the word. But no one says that you have to be smart. And someone said, but you are here, so of course you are smart. I mean, you are not stupid. You have a PhD, you have a career in science, you have everything. So, and sometimes it's something that we forget, all of us, right? And then you have also this thinking like in your head, like here, you are not, and eventually you are capable.
Yeah, no, this is the worst feeling ever, and this you will always feel. And I remember applying to whatever award, and then I wrote the text, and then I asked a friend of mine who is a journalist to revise the text, you know, and then I wrote about this imposter syndrome, and he told me, stop even saying that. Don't say that. You have to remove it from, you know, even the thought, just remove it from your head. But it's always there. I don't know why. And then I don't know. Sometimes I really have the feeling. I mean, if I speak for myself, I didn't know I wanted to be a PI, right? I just somehow kept in science because I love doing science, and it's the thing that I think about a job. So we want to do when you are a grown up, right? So science was my thing, but I didn't really apply the equation science equal being a PI, right? So I actually realized it very late, and this influenced a lot the fact that I have also this super big imposter syndrome because it's like, okay, if you didn't even think about it, so why, so how, why, what, right? So it's sometimes it's very difficult to deal with it, and sometimes you cannot really deal with it, right? So it's and for example, well, I don't know, I don't really have tools to try to convince myself of, you know, that what I have, I deserve it. And then I have it because I could, you know, reach it somehow. Sometimes I have this feeling that everything rolled somehow, and just I am where I am because I was on top of a mountain and it just rolled down. But actually it was the opposite, right? So usually it's going up, it's not going down, difficulty-wise as well.
Yeah, I agree with Sara. The very big driver for staying in science, and I think you need to love science, right? You need to be like excited about the things that you do, and I think it will take you eventually whatever, you know, path you take. But if there, yeah, this is, I think, something which is like the key point, you know, that keeps you going. Yeah, because there is like a lot of failures up and down. We criticize ourselves, and we need to be critical. I think in science we need to learn how as well to be critics, otherwise to ourselves, to others. And this also sometimes is kind of contradictory, what, you know, what is the imposter syndrome, right? And then we kind of face it, but it's the way how it works. But this is how we, you know, how we progress. And also I think it's important. So yeah, I have a question actually for Laura. Maybe you can tell to the students that are connected how you decided to stay in academia.
I actually had this question for you both, you and Sara. How you,
but
which
for
was
us,
the moment, right? Which
I
moment
think
did you say,
I think the most important transition is, you finish your PhD, you go to postdoc. I think this is where usually, I think there are statistics that actually says that, especially women, they kind of drop the percentage of women from PhD to postdoc. And then from postdoc to PI, it's like significant, with 3 stars or 4 maybe.
And if you are a mother, then forget about it almost, right?
Yeah. So Laura.
So in my case, I have to say, I mean, I also, as I said, I really enjoyed my PhD, scientific-wise and also personally. I mean, I had a lot of friends from my PhD, and I enjoyed a lot. But I was not sure if I wanted to continue. This is something that I think that a lot of people have this feeling when they finish their PhD. But I enjoyed, but I think I was also very lucky. So I actually finished with a very good curriculum, let's say. I had quite a bit, I mean, I think I had a lot of papers published, not first author, but, in the middle or second or whatever. So, and of course, I liked it. I mean, I love it. I love science. I love research. I love all this. But when I had to decide if I wanted to continue or not, I had a lot of friends that actually, they really loved science and wanted to continue, but because they didn't have a very excellent curriculum or whatever, they had to quit. So I kind of felt that I had I know it's kind of stupid, but I felt I had the responsibility, since I had a good curriculum and I could do it, to at least try it. And to be honest, that's why I did it. I mean, apart from the fact, as I said, that I like it, but this was kind of the thing that pushed me, right? That you have a good CV because you were lucky. Of course, I hard work, right? It's not that everything is because of luck. But I had kind of this, I don't know. I felt really bad for those people that could not pursue, their career because of not having very good papers published. So yeah, and I have to say I'm enjoying a lot. Yeah.
Very nice.
And what about you? Is there a moment in your life where you say, okay, I'm gonna be a PI? Or it just now you flow over the time and you apply and let's see what's happened?
Well, flow is complicated, right? Because the how do you say, the probability that you will find a PI position are not super high, right? So really you have to be convinced to go through this other round of frustration that will come from all the rejections, basically, right? Which is fine, right? So now, because at the CLG, right, that we will have to eventually leave after 9 years, the junior PI. So I heard when I started at CLG, another PI told me that for finding a senior position, he interviewed 100 times. And I was like, how is it possible? There are no 100 positions open in 1 year, Paul. So how can you interview 100 times to find a senior PI position? So that scared me a lot. But anyway, I was told, why don't you apply? And I was like, what? Yes. And then it was the director of the institute where I was before. And he told me, I think you have a nice CV. You have the whatever temperance, the character. You can do it. And it was like really cheering me. Like, really, you can do it. You can do it, because I didn't think I could do it, or I didn't even think about it, as I said before, right? So it was not in my mind. I was doing my science. My project was super nice. I loved it. I could have stayed like a postdoc forever. Also, I love the place. I love the institute. I could have stayed there forever, I think. Of course, it's not possible, right, as a postdoc, unless you become, you get another different position, more staff scientist or something like this. But I didn't think about it. And then when the director of the institute told me that, I really started thinking about it, but it was 2017, right? So it was like 1 year before getting the position at the CRG. This is the time that I thought about being a PI, so 1 year. And usually, so I was comparing myself with, you know, for example, German friend that I had, that they were working with me, that they knew since they were like 2 years old, that they wanted to be group leaders, right? And I was like, I don't even know what to eat
working
tonight.
mitochondria, no?
And working mitochondria. And then in my life, I really changed it. You know, I started with the PhD or the master when I was in Florence in epigenetics, then, well, first in macrophages, then in epigenetics, then I did the second PhD was in mitotic regulation, and then the postdoc in drug screening. So I did a mix of things that it wasn't really following any path, which at the end, it was nice because I could merge everything together. And now, for example, in the lab, super cool because, you know, we find metabolic enzymes that we can inhibit in the nucleus, which are related to mitosis progression. And, you know, I can put everything together in my mind and do the puzzle. No, it's super, super cool. But I didn't really have this path very clear in my mind. So I think it was really when he told me, I think you can do it, that I trusted him and I decided to do it.
Yeah, I think it's very important to have mentors, you know. So this is what actually I said to everyone. So your mentors can be your supervisors, but also the best mentors are actually your friends, right? So because they see your qualities and that you actually maybe cannot see, you know, so they kind of tell you this, you know, kind of raise you these questions. So you should do it, you know, and you're like, oh, me? Why me? I never thought about this, no? But then actually, that's why I think for PhD students, postdoc, even for us, like young PIs, I think it's so important to have mentors because, you know, they can make, you know, these critical moments and questions and challenge you a little bit, see some things that maybe in your little world you cannot really see, you know. So yeah, get a mentor.
But did you already knew that you wanted to be a PI, Anna? I mean,
well, it's more, no, it's more similar to Sara. So actually it was my friend that started to send me advertising. I said, oh, I have to go back to Barcelona because of family reasons. And she started to send me all these advertising for the PIs. And I was like, okay, why she sent me, you know, I said, if you see some job openings or something, you just send me advert. And she's like, keep sending me the advertising for the PIs in Barcelona. And I was like, why is she sending this to me? I said, oh, maybe, you know, if you see something else, no, you go for this, you know. So yeah, and then then I started to think, you know, that why not? You know, and then you start to think and you build up a little bit idea and you start to applying and you start talking to the mentors and is what they say, yeah, just go for it. And yeah,
I think it's very important actually to see that someone else, you know, sees that even though you didn't see that. I think this is very important because if someone else believes that you can do it, even if you didn't share this idea of you really wanting doing this, right? Then it makes you feel very confident that you can actually do it, right? It's that's very nice. But this is true for any kind of job, right? That you not just, you know, because if someone tells you now, you know, you go to industry, I will freak out literally because I don't even know where to start, right? So it really makes a difference having someone, a friend, a colleague, whatever, your partner, whoever, right? That tells you, sure, you can do it. I can see this potential, right? I have no doubts that, and I think it's fundamental.
Yeah, and especially for us that we did like so many, like during the PhD, you have so many skills that you can convert to like a lot of like applications, and you can apply to industry, you can apply for scientific writer, you can, because you know,
and sometimes you're not aware of that, right?
And you're not aware, you need someone actually to tell you, yeah, but you did this, and this is this description, because sometimes we don't see it, I think. So, yeah.
Yeah, honest, when I was thinking about if I wanted to do a postdoc or not or whatever, I was also looking for this kind of industry positions, blah, blah, blah. And they asked for some skills that I was like, okay, this is not me for sure. And someone came and said, of course, you fit on this. Why? I mean, because in your PhD, you did this, that could apply to this. Okay, maybe you're right.
Yeah. And actually, sometimes they ask like 4 years of experience and you think that you have never worked, but actually the PhD is working. I mean, it's like a work experience in that sense. So yeah, sometimes you don't even realize, like, because we are like looked as students, but we are actually working. Like it's a job. It's
of course.
And
don't let anyone tell you the opposite, because
no.
This is horrible.
And I think you learn, the most important thing is not to be afraid. So the beginning, in my first year, I was always afraid of learning a new thing. And I was, okay, if I have to learn like this new technique, I was seeing like a big wall in front of me, like, oh my God, I have to now spend a lot of time learning a new technique. But this over the years, as Julia was saying, it becomes easier. Like, oh yeah, I have to learn this new thing. It's fine. In one day I got it. And this confidence that gives you is super important.
And yeah, you become more faster learner. You become more faster learner how the years progress. So usually in the beginning of PhD, it takes that amount of time. But when you start from a postdoc, it's like the changes are more rapid. But
definitely.
Bye.
Wow, it has been a pleasure to listen to you all. And we always say that this part is very, very enriching because we share a lot of experiences. Definitely mentoring is super important. Although Anna's friends show, it's more like a pusher than a mentor, like giving you all the PI offers. But anything is good. So thank you again for organizing this elevator pitch. It was amazing, great science, and you all have a very bright future ahead of you, the young ones, and the PIs. Well, what can I say? It was very, very nice. I don't know if Maria can join us for the goodbyes.
Yes, just to say goodbye. I really like all the results that you have shown and the talks. I mean, congratulations to all of you. You have great projects and great mentors too. So lucky. And just to thank you again for hosting this elevator pitch.
Yeah, thank you all. And we'll see you in Malaga.
Okay. Thank you.
Thank you.
Bye.
Bye.
Bye. Bye, everyone.
Bye.
Bye.
Bye. Ya estamos fuera del directo.